Using a pen, mark B on the side of the slide which you are going to smear the bacteria culture. With the clothespin securely attached to the opposite end of the slide, move the slide inside the flame from side to side for approximately 5 seconds to heat fix the marking to the slide.
With a clean inoculating loop, take a small sample of a known gram-positive bacteria culture and thinly smear it on the left side of the slide. Put the inoculating loop inside the bleach solution afterwards to kill the bacteria culture on the loop. Likewise, with a clean inoculating loop, take a small sample of a known gram-negative bacteria culture and thinly smear it on the right side of the slide. Place this inoculating loop in the bleach solution as well.
Heat fix both bacterial smears onto the slide for about 10 seconds. Heat fixation kills the bacteria, allowing them to stain better. This also allows the cells to stick tightly to the slide.
Place a few drops of crystal violet dye on each bacterial smear. This dye is composed of basic or positive ions that adhere to the negative surface charge of the bacteria. Wash this dye off with water for about 2 to 3 seconds, and then place a few drops of iodine solution on each bacterial smear and wait for approximately 60 seconds. The iodine acts as a mordant that ensures that the violet dye adheres to the cell and forms crystal violet iodine complex. Wash this dye with water as before, and afterward blot the slides dry.
Flood the slides with a continuous stream of ethyl alcohol for about 10 seconds with the slide, held at a right angle or until no more dye comes off the slide. This ethyl alcohol decolorizes the smear by dissolving lipids found in the cell walls of gram-negative bacteria, dehydrating the bacteria and thus allowing the crystal violet iodine complex to leak out of the gram-negative bacteria.
Flood each bacterial smear with saffron dye for about 20 seconds. This dye will act as a counterstain, meaning that since the gram-negative bacteria do not have the outer lipopolysaccharide layer anymore, they will easily absorb this red dye, whereas the gram-positive bacteria won't and will instead maintain their purple color. Rinse the slides with water for about 10 seconds and air dry.